Welcome to today's Arctic Research Seminar. It's hosted by the Arctic Research Consortium of the United States, or ARCUS. Today's team includes Stacy Stout, Kubajenda, and me, Betsy Turner-Bogren. Our seminar, entitled Marine Predators as Sentinels of Environmental Change in Arctic Ecosystems, will be presented by Dr. Emily S. Choy from McGill University and Environment and Climate Change Canada. A link to an online survey will be available at the end of today's webinar. We'd appreciate your feedback as well as suggestions for future webinars. We have a couple housekeeping items before our presentation begins. You have two viewing options available today, gallery view and speaker view. Gallery view will allow you to see the webcams of other people on the webinar, and speaker view will allow you to view only the presenter's webcam and presentation. To switch between those views, use the button in the upper right-hand corner, which is indicated by the red arrow on your screen. Zoom controls, including chat, can be found by hovering your mouse over the bottom of your Zoom window. You're invited to chat with other webinar participants at any time. To do that, click the chat icon at the bottom of the screen and select the name of the participant with whom you wish to chat. There's also an option to chat with everyone participating in the webinar. Questions will be addressed during the question and answer session following the presentation. If you're joining by Zoom, please type your question into the chat window at any time. I will collect those and then read them aloud during the Q&A. If you're joining by phone, you will need to raise your hand by pressing *9 and unmute yourself by pressing *6 to ask your question aloud. I will give you a reminder of these guidelines before we start the Q&A. If you have any technical problems today, please contact Stacy Stout or Kuba Jinda. Stacy's email is Stacy, S-T-A-C-E-Y, @arcus.org, and Kuba is K-U-B-A @arcus.org. Before we begin, we'd like to thank the National Science Foundation Office of Polar Programs for their financial support to ARCUS and to this seminar series. Now I'd like to welcome our speaker. Dr. Emily Choy is a postdoctoral fellow in the Department of Natural Resources Sciences at McGill University and the Environment and Climate Change Canada. Her research is supported by the Natural Science and Engineering Research Council of Canada, or NSERC, and a L'Oreal UNESCO for Women in Science Research Excellent Fellowship. Dr. Choy is studying the physiological response of thick-billed murres on Coates Island, Nunavut, to Arctic climate change, specifically the effects of change in prey availability on their energetics and warming temperatures on their physiology and behavior. She completed her PhD in biological sciences at the University of Manitoba in Fisheries and Oceans, Canada. Her investigations were on beluga whales as sentinels of environmental change in the Beaufort Sea ecosystem in partnership with communities in the Inuvialuit settlement region of the Northwest Territories. Dr. Choy completed her Master's of Science degree in biology at the University of Ottawa, studying the ability of seabirds to transport contaminants to coastal food webs at Cape Vera, Devon Island, Nunavut. Dr. Choy was a scientist on the Victoria Strait expedition and a recipient of the Erebus Medal. She is a fellow for the Royal Canadian Geographical Society, a scientific advisor for the W. Garfield Weston Foundation's Northern Committee, and a council member for the Association of Field Ornithologists. Dr. Choy is very passionate about science outreach and is currently partnered with Earth Rangers in their Northern project to teach kids about conservation of Arctic wildlife. Please join me in welcoming our speaker, Dr. Emily Choy.
Thank you very much, Betsy. I'll just share my screen. So good morning, everyone. I'm very excited to be here today. Thank you very much for having me. So for today, I'll be sharing with you my research on marine predators as sentinels of environmental change. I'll be starting with my PhD work on beluga whales and then finishing off with some of my postdoctoral work on seabirds. Before I begin, I would just like to say that although I'll be sharing with you my PhD results, it was part of a larger collaborative project with partners across northern communities, Fisheries and Oceans Canada, and several universities who all helped to make my research possible. There are several characteristics that make beluga whales an ideal sentinel species for monitoring Arctic change. They're the most abundant Arctic toothed whale with a circumpolar distribution. So they're found across the Arctic. They're highly specialized for Arctic environments, For example, they lack a dorsal fin, which helps them to navigate under sea ice. They also have thick layers of blubber, which serve several purposes, including thermoregulation, but also buoyancy. They're long-lived with a low reproductive rate. So there are whales within my study population that can live beyond 60 years of age. And females tend to have one, sometimes two calves every 3 years. And finally, they use many different habitats. Beluga whales will use sea ice habitat, offshore habitat, estuarine habitat, coastal habitat, which is important for monitoring a complex ecosystem such as the Beaufort. Now, specifically, the Beaufort Sea beluga population is one of Canada's largest, with an estimated 40,000 individuals. Beluga whales spend their summer in the Canadian Beaufort Sea, so the whales arrive around late May to early June. The females give birth to calves around early July in the warmer waters of the Mackenzie River estuary. The whales spend their summer feeding in the Amundsen Gulf before returning to their winter habitat in the Bering Sea around late September, early October. Beaufort Sea beluga whales have a strong habitat association with sea ice. So males and females are sexually dimorphic, and based on telemetry studies, large males tend to select offshore pack ice, whereas females with young calves tend to select open water habitat near the mainland. They're also highly specialized divers. So beluga whales can dive between 400 to 900 meters, sometimes beyond 900 meters, and it tends to be the larger males that make these dives, which are believed to be for foraging purposes. And finally, Beaufort Sea beluga whales are an important traditional food to communities across the Inuvialuit settlement region, and beluga whales are also harvested across the Arctic. Now, recently, there was a 20-year decline in inferred growth rates within the Beaufort Sea beluga population. So from about 1988 to 2008, beluga whales were getting smaller with age. And this decline corresponded with a number of declines in body size and condition to several marine predators within the Beaufort Sea, such as Arctic char and ring seals. Now, although this decline is believed to be due to environmental change, the exact mechanisms are currently unknown due to a lack of baseline information on the diet, the physiology, the health, and general biology of the whales. So the overall objective of my PhD research was to examine interannual variation and potential environmental factors affecting the prey, body condition, and physiology of Beaufort Sea beluga whales for insights into changes within the ecosystem. Now, in order to address my objective, I had several questions. So I looked to see how beluga prey varies with environmental factors as well as habitat use among the different habitat use groups of belugas. And then secondly, I also looked to see how beluga diving physiology and foraging, potentially foraging, is affected by changes in body condition, body size, as well as prey availability. Now, my research took place in the Inuvialuit settlement region of the Northwest Territories, and I spent 3 summers on Kendall Island, just off the coast of Tuktoyaktuk. But I worked in collaboration with several communities and camps across the Inuvialuit settlement region, such as Browns Harbor near Polytuck, Hendrickson Island, as well as East Whitefish. Now, my research was part of a community-based monitoring program in partnership with Inuvialuit communities across the Northwest Territories. Now, this program is co-managed by the Fisheries and Joint Management Committee, as well as Fisheries and Oceans Canada, and has been running since the 1980s. Beluga whales are an important traditional food to communities in the Inuvialuit settlement region. They have been for many years. So every year there is a hunt in which a small number of whales is taken from the population. And this hunt has been found to have no impacts on the demographics of whales, according to studies. So the whale population in terms of numbers is currently healthy. Beluga meat is extremely nutritious. It's very high in vitamin D, vitamin C, vitamin E, as well as omega-3 fatty acids. And there have been several studies that have shown that traditional foods such as beluga muktuk are much more nutritious than store-bought foods, which are often expired by the time they reach the Arctic or the Canadian Arctic, and often extremely expensive. So this is a partnership with communities. There's a lot of co-production of knowledge. For example, that decline in inferred growth rates that I showed, before that was even published, the communities have been telling scientists that the whales were getting smaller. And then finally, if it wasn't for this partnership, we wouldn't know as much about the Beaufort Sea beluga population as we do today, and I would not have had any samples for my thesis. So just to give you a sense of how this partnership works, I prepared some slides. In each of the hunting areas, there is a beluga monitor, and it's the job of the beluga monitor to basically keep track of the numbers of whales harvested, take measurements of the whale, as well as a subset of samples for tissue and health, for health and contaminant analysis. So the research team that I was a part of would work with the beluga monitor. And what would happen is when a hunter lands a whale, they would radio the beluga monitor, who would then meet the hunter at their campsite. Now, although I was on Kendall Island, there are several islands within the area. So in this picture, this is a picture of a hunter who is hauling in a whale. And this could take anywhere from 30 minutes to several hours, because they would have to move slowly with their boats. Next, once the whale is brought to shore, the community members come together and basically haul the whale out of the water. And again, this is a remote location, so there's no access to vehicles or machinery. This is basically done with rope and human strength. And these whales are heavy. They could weigh over a ton. And then once the whale is brought on shore, then the monitor will take their initial measurements of the whale. So this is a picture of beluga monitor John Day and myself measuring the length of the whale. The monitor will also measure the girth, the blubber thickness, the flipper size, the tail size. And then once the monitor has finished their measurements, then usually the families of the hunters will start to cut up the whale for their food. And it's usually at this point where I will ask permission to take a tissue sample for my research. Now, as you can see, there's nothing but kids, there's nothing but families at these camps. Usually a family will take no more than one whale that will last them throughout the year. And then also as part of this program, there's also many local youth involved. So this is a picture of Kendra Tingniak, who was a field assistant on Kendall Island and in charge of the blood work one year. And she's gone on to be an Inuit research advisor for ArcticNet. Now, the communities are mostly interested in the blubber as food. So what they'll do is they'll cut the blubber into strips, and then they will hang it out to dry in the sun. So this is beluga blubber or muktuk being dried, and then it'll be further prepared in stews or cooked in other forms. And then secondly, they'll also consume the mussel or nitku, which means dry meat. So this is mussel being dried, and this is dry fish. So when I was living with the communities, I consumed mainly traditional foods with them as well. Now I just have a few pictures of life, camp life on Kendall Island as part of a community-based monitoring program. Again, we do a lot of consultation with communities during the winter, during our off season in terms of the field. We're usually up north giving talks for hunters and trappers organizations as well as local schools. But also when we're at the camps, we're always communicating with the families and the hunters about our work and also participating in activities with them as well. So this is a picture of me learning how to bead. That's a very important cultural activity within the Northwest Territories. This is a picture of us playing a game of Snert. Snert is a very popular card game in Inuvik. It's a game of speed in which you're trying to get rid of all of your cards as fast as possible using runs. And again, when we're at these camps, there's no access to electricity or Wi-Fi, so we tend to play a lot of card games to stay entertained. And finally, my favorite, we would play baseball. So once in a while, the communities would, or the families would radio each other and play a game of baseball, which is a very popular sport in the Northwest Territories. And then we would all meet on an island to play. Now, although this looks like it's taking place in the middle of the afternoon, these games would tend to start around 11 p.m. at night and continue to about 3 in the morning. So on Kendall Island during July, we do experience 24 hours of daylight. But it's thanks to this wonderful partnership that I was able to acquire almost 200 tissue samples from my work on beluga prey and diet, and almost 80 tissue samples from my work on beluga physiology. So to continue this, I'm going to be presenting my work that was recently published in the Marine Ecology Progress series on beluga prey. And so the overall objective of this part of my research was to examine the relative proportional contributions of the most abundant prey species in the Canadian Beaufort to the diets of beluga whales. And I did this in partnership, well, not only with communities, but also with the Beaufort Regional Environmental Assessment Program, which was a big program run out of Fisheries and Oceans Canada to survey the biodiversity of fish and invertebrates in the Canadian Beaufort Sea. Now, in order to address my objective, I had a few sub-objectives. So first, I determined whether 14 prey could be differentiated based on ecological tracers of diet. And I used 2, so I compared 25 fatty acids, so fatty acids transferred exclusively from prey to predator. I compared the fatty acids in the inner blubber of beluga whales with that of the prey. And then secondly, I also used stable isotope ratios. So I compared stable isotopes in the liver of beluga whales with whole body stable isotopes in the prey. Now you might be wondering why I had to use ecological tracers, because I had access to beluga stomachs at the camps. Well, the beluga stomachs tend to be empty. We're not sure why, but the hunters tell us that when they're being chased, the whales will actually spit out their food. Secondly, I examined the hypothesis that beluga whales prefer lipid-rich prey. So again, one of the predictions of Arctic change is a potential decline in prey quality. And then lastly, I reconstructed the individual diets of 178 beluga whales using a mixture model, quantitative fatty acid signature analysis in R. So in terms of my results, my first objective, I found that I could effectively differentiate among my prey using fatty acids. So these are 2 correspondence analyses of the fatty acid compositions of individual fish and invertebrates prey. So this here, as you can see, I just basically put, as you can see, I found there was a gradient between more the fatty acid patterns of more pelagic species versus more benthic species. And if I overlaid the fatty acid patterns of my beluga, I found that they were more similar to fish species such as halibut, Arctic cod, and capelin versus benthic species such as sculpin. So I ran the same analysis using my stable isotope ratios. I found that stable isotopes were able to differentiate among most prey, but not as effectively as fatty acids, likely because I'm only using 2 tracers, whereas for the fatty acids, I use 25. Just to give you a sense of what stable isotopes are, nitrogen is used to infer trophic position. So it tends to increase 3 to 5 per mil with every trophic transfer, whereas carbon is used to differentiate among baseline primary productivity. So within an ecosystem, I mean, if I expect beluga whales to have a diet mainly of pelagic prey, I expect that their carbon signals will overlap. So these are the labels of my different prey types. And in terms of beluga whales, I found the carbon signature within the liver did not match the prey. But of course, unfortunately, there's not a lot of information on turnover rates in terms of different tissues of whales. And because they had recently migrated a couple months before, it's possible that the carbon signal of the liver might reflect prey in the Bering, which often have a much higher carbon signal than prey in the Beaufort. But based on the uncertainty of these results, in terms of my mixture models and diet reconstruction, I decided to stick with my fatty acids and not my stable isotopes. So in terms of my next sub-objective to look at whether beluga whales prefer lipid-rich prey, I compared the lipid content of my different prey species and found that they did overall differ in lipid content. And in fact, more pelagic fish, such as capelin and Arctic cod, had the highest lipid content, in particular relative to benthic invertebrates, which had the lowest lipid content of all species. Now, finally, I ran a mixture model to reconstruct the diets of 178 individual beluga whales. So I used, again, 25 fatty acids with 12 different prey classes. So I ended up, based on my correspondence analysis in Permanova, grouping my decapods together because they had very similar signatures. And I found that overall, Arctic cod was the most dominant prey of beluga whales, which is no surprise that historically they've been the most dominant prey and they're the most abundant fish in the Beaufort, followed by capelin. But I found that beluga whales in general, Beaufort Sea beluga whales in general, had a diverse diet. They fed on Canadian eelpout, Greenland halibut, kelp snailfish, and decapods as well. Now, when I compared different habitat use groups, the diets of different habitat use groups of whales. So I ran a Permanova with different habitat use groups as a predictor. So from females, I grouped them together. For males, I grouped them into 3 habitat classes based on previous work by Lizzetto, my supervisor Lisa Lizzetto, so large versus small whales. But overall, I found that large males consume the highest percentage of Arctic cod as well as Greenland halibut and the least capelin relative to all males. And also, I also found that females consume the highest percentage of capelin and the lowest percentage of Arctic cod relative to all groups of males. In my models, I also found that year was a significant factor. And in fact, that beluga whales in 2014 consumed the least Arctic cod and the most capelin relative to whales from all previous years. So just in summary of these results, I found that beluga whales prefer lipid-rich prey, feeding primarily on Arctic cod. Oops. Diet estimates varied with habitat use. So females consume the least cod and the most capelin, whereas males consume the most cod, large males consume the most cod, the most Greenland halibut, and the least capelin. And this was supported by a few things. So both our knowledge on sex and size differences based on habitat use. So again, females tend to use more, particularly with young calves, tend to use more of the coastal open water habitat than large males, as well as the BRIA survey. So in terms of the fish and invertebrate surveys, capelin were found exclusively in the coastal habitats where the females reside, whereas Arctic cod was found across the Beaufort, but in particular, large Arctic cod and Greenland halibut were found in the deeper water, the greater depths where males can dive to. And secondly, I found that diets estimates varied with year, and beluga whales consumed the most capelin and least Arctic cod in 2014. And this was also supported by both the BRIA fish survey. So in terms of Arctic cod was in its lowest abundance or biomass in 2014 relative to 4 previous years. So from 2010 to 2014, 2014 was the lowest. And in particular, 2014 was a very odd year for Beaufort Sea belugas. So based on our work with communities, it was reported that beluga whales had actually swam up the Mackenzie River in 2014 to Inuvik. They actually died, so they weren't healthy, but that was a first. And beluga whales were also harvested for the first time in over 30 years in the community of Lulahaktuk. And these whales had stomach contents and they were filled with sand lands. So we believe that because it was a low cod year, beluga whales, Beaufort Sea beluga whales might have had to depend on alternative prey species, such as capelin and sand lance. So I'm just going to follow up with some of my physiology work. So again, going back to this graph of the decline in inferred growth rates with over time in Beaufort Sea beluga whales. One of the questions I had was a PhD student was, could this impact diving ability? Because in toothed whales, the bigger you are, the deeper you dive. So there's a strong correlation between body size and dive depth in Arctic and odontocetes whales. So why is bigger better in terms of diving? Well, it all has to do with how oxygen stores scale with body size in elite diving mammals. So if you're an elite diving mammal, you'll store most of your oxygen in your blood. You'll have very large blood volumes and high concentrations of an oxygen transport protein known as hemoglobin. Secondly, you'll have large muscle masses or large muscle oxygen stores. So you'll have large muscle masses, but also have high concentrations of an oxygen storage protein known as myoglobin. And lastly, unlike us, you'll store the least amount of oxygen in your lungs. So having large lungs is a bit of a liability when you're diving over 900 meters. You might suffer from the bends or decompression sickness. So elite diving mammals will tend to have small lungs relative to their body size, and they will actually collapse their lungs upon descent. Now, if you know the concentration of oxygen in these three tissues and the relative volumes, you can actually estimate the total body oxygen stores within a marine mammal. Now, if you divide this by the metabolic rate, which you can estimate through allometric equations based on body mass, you can actually estimate the aerobic dive limit, or the theoretical time a marine mammal can dive aerobically with oxygen underwater. So the overall objective of this part of my research was to get baseline information on the dive physiology of whales, examine the relationship between physiological parameters of oxygen stores and their relationship with indices of condition and body size, to examine if declines in inferred growth rates could impact foraging ability. And so this research was recently published in the Journal of Experimental Biology. A secondary objective was to examine the role of the spleen in beluga whales. So many elite diving faucids will have very large spleens relative to their body size. So a good example is the whittle seal and the southern elephant seal. And it's believed that this spleen will act as a blood storage reservoir. So when these seals dive, they'll contract their spleens and increase their blood volumes. The role of the spleen has been less studied in cetaceans. So I sought to see whether it could serve a purpose in diving. So thanks to my partnerships with communities, I was able to obtain 77 tissue samples from whales, in which I measured the following physiological parameters. So I looked at blood hemoglobin and hematocrit. Hematocrit is the percent of blood cells in a given volume. Myoglobin and buffering capacity in the leucas dorsi muscle. The leucas dorsi is the main diving muscle in marine mammals. It runs along the back. And buffering capacity is a measure of how well a muscle functions under anaerobic conditions. And contracted spleen mass. And then finally, using linear models, I examined the relationship of these parameters with body mass, condition, age, and sex. And then I estimated the total body oxygen stores and dive limits of individual beluga whales. Now, in terms of my body condition indices, I used two. My first was a body condition index based on the residuals of a model fitted to maximum half girth. This had been previously used for bowhead whales and was found to be associated with the open water period and period of peak prey availability. And I had compared these with another indice in Beaufort Sea beluga whales and found that it was most associated with dietary traces of Arctic cod, the open water period, as well as period of peak prey availability as well. And secondly, I used a maximum half girth to length ratio as a body condition index, which is a more common indice in mammals in general. Now, in terms of my results, I found that beluga whales, or Beaufort Sea beluga whales, had one of the highest myoglobin concentrations of all marine mammals. So 83.9 milligrams per gram, corrected for impurities such as blood, 77.9. But the first value, 83.9, tends to be what is reported in the literature. I found that Beaufort Sea beluga whales had high hemoglobin concentrations, and they had fairly high buffering capacity. So they're an endurance swimmer. Now, when I compared these values with other marine mammals in the literature, I found that they were most similar to the narwhal. In addition, I also sequenced the myoglobin gene of beluga whales and found that there was a 99% similarity in coding sequence with narwhal, which is no surprise, they're from the same family, but Dr. Terry Williams has done a lot of work on narwhal physiology and believes that because of their extreme physiology, so they're very slow endurance swimmers, they can dive over 1,000 meters for Greenland halibut, but that might make them vulnerable to rapid climate change or environmental change. So based on these results, we believe that beluga diving physiology is more similar to narwhals than previously anticipated, which may make them vulnerable to rapid change. Now, in terms of my models, for blood oxygen storage parameters, I found that body condition, and both indices modeled separately, was the most significant predictor of blood oxygen storage parameters. So hematocrit was best predicted by body condition alone. Hemoglobin was best predicted by body condition, age, sex, and their interaction. But overall, body condition was the most significant predictor. So I found that beluga whales with the highest condition also had the highest percent hematocrit and the highest hemoglobin concentrations. For muscle myoglobin, I found basically the same model. So body condition best predicted muscle myoglobin. So beluga whales with the highest condition also had the highest myoglobin concentrations. In terms of buffering capacity, I found it was best fitted by the null model. Buffering capacity isn't an indication or related to oxygen storage. It's only related to endurance. And for spleen mass, I found it was best fitted by age and mass, but I found the spleen only comprise about 0.02% of the total body mass of whales. So based on the small size of the spleen relative to the total body size of the belugas, we believe it does not serve a role in diving. Now, in terms of the total body oxygen stores of all of the whales, 77 whales, and their relative distribution, we found that the mean total body oxygen stores of whales was 58.7 milliliters per kilogram, which is similar to the short fin pilot whale, another elite diver. And beluga whales stored most of their oxygen in their blood relative to their muscle and lungs, which is a similar profile to other prolonged diving mammals. Now, when I compared males and females, I found that although males and females had a similar percent distribution in total body oxygen stores, so they stored the same percent of oxygen in their blood versus their lungs versus their muscle, when I took into account differences in body size between whales, I found that because males are significantly larger, males had significantly larger total body oxygen stores than females, which may explain why in historical telemetry studies, they've been shown to dive to greater depths than females. Now, one of the questions we had was, because we found these significant relationships between body condition and blood and muscle oxygen storage parameters, Could this impact total body oxygen stores? So what we did was we estimated the total body oxygen stores for 77 beluga whales. But in order to look at the impact of body condition alone, we controlled for body mass. So we assumed all the whales were the same body mass. So we used the mean. And so using allometric equations as well as the measurements for hemoglobin and myoglobin, we estimated the muscle oxygen stores for individual whales, blood oxygen storage for individual whales to get total body oxygen stores. And so we just assumed, we calculated lung oxygen stores based on the body mass, so they all had the same. In addition, we also compared the total oxygen stores with the whale with the highest versus the lowest body condition index, assuming they had the same body mass. What we found that overall, the beluga with the lowest body condition index had 12% lower muscle oxygen stores, about 27% lower blood oxygen stores, which equated to about a 3-minute difference in dive time in comparison to the whale with the highest condition. So 14.3 versus 17.4 minutes. Again, this is a very conservative estimate because in reality, they don't have the same body mass. But based on these results, we believe that the impact of body condition on muscle and blood oxygen stores are cumulative, which may affect diving ability. Now, how could this affect foraging ability? Well, it all has to do with the differences in habitat associations among fish in the Beaufort, in particular with depth. So there's different fish communities based on the nearshore and offshore environment of the Beaufort Sea, but also with depth, in particular with the main prey of beluga whales, which is Arctic cod. So Arctic cod is found in its greatest biomass between 350 to 500 meters. But Arctic cod also displays a very significant size depth gradient. So year-old or young of the year Arctic cod tend to be near the surface of the water column, whereas the older, larger Arctic cod tend to be at the greater depths. So based on these results, we believe the beluga whales with lower condition and therefore lower oxygen stores may not be able to access the largest and greatest biomass of Arctic cod. So in summary, Arctic belugas or Beaufort Sea belugas are specialized for prolonged diving and navigating under sea ice, similar to narwhals. And declines in condition may affect oxygen stores in belugas. Therefore, whales in better physical condition may perform better under stressful circumstances, such as evading predators and ice entrapments. And we believe this could be a vicious cycle in which declines in condition, which could be caused by changes in prey availability, impact the dive time or aerobic dive limits of the whales, which impacts their ability to forage for their prey, which might further worsen their condition. And this was also recently featured in an infographic by whales and pineapples and whales. So finally, I think I'm going to move on to some of my postdoctoral work on seabirds. So similar to beluga whales, there's several characteristics of thick-billed murres that make them an ideal sentinel species for monitoring Arctic change. They're the most abundant seabird in the Canadian Arctic with a circumpolar distribution. So there's colonies in Svalbard, Norway, Iceland, Greenland, Russia, Alaska, throughout the circumpolar north. They're highly specialized for Arctic environments. So they breed and forage exclusively in waters near waters 8 degrees Celsius and lower. So are only found in the high latitudes. They're long-lived with a low reproductive rate. So in 2019, the oldest murre we studied was 38 years old. And once murre parents or breeding pairs reach about 3 to 5 years of age, murre breeding pairs will raise 1 chick per breeding season. And they use many different habitats. Obviously, they nest on cliffs on the coasts. They forage in the offshore environment, and they're also deep divers, so they can dive up to over 100 meters. Now at Coates Island in Nunavut, Canada, there's a colony of about 30,000 breeding pairs that nests on the cliffs of Coates Island. So these murres arrive around mid-May to early June to Coates Island. They begin to lay their egg, their one egg, and mid-June to early July. And then once the chicks hatch, they'll fledge usually the first or second week of August. Now, although the birds will spend their summer on Coates Island, they actually spend the winter off the coast of Newfoundland and Labrador. They're a highly specialized diver with a high cost of flight. So thick-billed murres can dive up to 150 meters in a matter of minutes, but they also spend about 4 hours a day flying during the breeding season. And in birds, flying and diving are an energetic trade-off, and it's very difficult to do both. And a good example, the perfect example of this is the penguin, which is an amazing diver, but has lost its ability to fly over time. So murres do both, but as a result of this, they have the highest cost of flight of all vertebrates, so birds and bats. And finally, they are an important traditional subsistence harvest to Inuit communities in Greenland, but they're also harvested in Newfoundland and Labrador. Now, recently, there have been declines in murre populations across the Atlantic. So in Greenland and Iceland. Now, although the Canadian populations have remained relatively stable, there have been several changes within the Coates Island colony. So over the past 30 years, the diets of murres at Coates Island has shifted from about approximately 50% Arctic cod to 50% capelin with the warming of Hudson Bay. Now, this shift in diet is believed to have caused a decline in chick growth rates. So murre parents will bring back one fish at a time to their chicks. And because of the smaller body size of capelin relative to cod, it's believed that this shift has caused a decline in growth rates based on long-term monitoring data on chick growth. Secondly, while the ice-free period around Coates Island has advanced by about a month, the peak period of prey, so the ice-free period, which is also the peak period of prey availability, has advanced by about a month. The murre breeding has only advanced by about a week. So there's a mismatch between the peak period of prey availability with the peak period of highest energy demands of the murres, which is during chick rearing. So one of the objectives of my postdoctoral work is to examine the indirect effects of prey shifts on the energetics and foraging behavior of murres. And in addition to working on murres, I'm also working on black-legged kittiwakes on Middleton Island off the coast of Alaska. And so I'm using a lot of novel technologies. So I'm first looking at heart rate as a proxy of oxygen consumption, as well as fine scale energetics. And I'm also using GPS-accelerometers to examine variation in behavior and activity rate in regards to heart rate and energetics of birds. And lastly, because at Coates Island, Arctic warming is bringing a number of changes, increases in shipping, as well as contaminants, I'm examining the impact of multiple stressors of birds by creating risk maps to map areas of where the birds are obtaining prey availability, but also concentrations of mercury and perfluorinated compounds as well. So in 2019, I completed a small pilot study on heart rate in both kittiwakes and murres. So heart rate temperature loggers were implanted in both murres and kittiwakes for a period of about 3 to 4 days. And then to make sure that if these loggers had any impact on the bird's behavior and energy levels, we also attached GPS-accelerometers to a subsample of kittiwakes and compared them with controls. So six kittiwakes that had implants and GPS-accelerometers and 6 that only had accelerometers. Using the accelerometers, the behavior of the kittiwakes was classified into 3 categories. So using hidden Markov models. So on the colony, on the water, and in flight. And then we estimated daily energy expenditures using activity-specific metabolic rates available from Jodas et al. So basically, we estimated the energy of the birds on the colony, on the flight, on water during the course of the day in order to get total daily energy expenditure. So in terms of our preliminary results, we found that in terms of heart rate, kittiwakes had a much higher heart rate than murres at about 370 beats per minute versus 270. It's not surprising because the kittiwakes are about half the body size of murres, but they had a much greater variation in body temperature. We're hoping to further examine this. Again, it might be because kittiwakes, murres are a diving seabird, and they've been found to have a very constant body temperature when they dive, whereas kittiwakes do not dive. But we need to follow this up with more studies. But in terms of the comparison between control versus implanted birds, we found that the heart rate loggers had no impacts on the foraging movements and behavior states of control and implanted birds. So these are two maps. We used residence in space and time models in order to classify the behavior of the birds into three states. So black is in transit state, blue is active foraging, and red is resting. And we found no difference in the distance traveled versus the amount of time spent in these three states. And then in terms of our behavior classifications using our accelerometry data, we found that there was no difference between the behaviors of control and implanted birds. So the birds both spent the same amount of time in flight, on water, on colony over the course of the deployment period. And they also had expended the same amount of energy. So we're hoping once COVID subsides to continue this work, do some longer term deployments, as well as finer scale calibrations between heart rate and GPS accelerometers in order to look at differences in energy expenditures between the specific bird behaviors. So the second question I am looking at or asking is, are murres vulnerable to Arctic warming? So murre breeding pairs will actually take turns spending 12 to 24-hour shifts incubating their nests. And there have been reports in the past of murres dying on their nests due to heat stress on moderate temperature days, so days around 22 degrees Celsius. So the overall objective of this part of my research is to examine the heat tolerance of murres. And we predict that as a cold adapted seabird, murres would exhibit signs of heat stress at low temperatures relative to more heat tolerant and desert birds, and also a very high cost of thermoregulation. So in order to cool themselves. So in order to conduct this work, I did this work in 2019, and I used flow-through respirometry to measure resting metabolic rates, evaporative water loss, as well as evaporative cooling efficiencies in murres at increasing temperatures from about 16 to 39 degrees Celsius. So I looked at these measurements as basically indicators of heat stress. In addition, I also monitored cloacal body temperature in each of the birds. So this was my setup. So this is a murre in a metabolic chamber. And then surrounding the metabolic chamber is a heating box. And so how this basically works is there would be air would flow into the metabolic chamber at a constant rate, and it would be scrubbed of both carbon dioxide and water. And then this air, the air in which the murre is in, and of the box the murre is in, would flow out and be measured for oxygen concentration as well as water vapor by a field metabolic system. So the temperature would be controlled by both a MobiCool cooler, so there's a MobiCool that would heat it at the top, as well as heating pads. And the measurements would start in the thermal neutral zone of the bird, so around 16 degrees Celsius, and would increase by about 2 to 3 temperatures. And so we would take measurements for about 30 minutes while the birds are resting. In order to make sure that the birds were resting and they weren't in any distress, there was a video camera at the top So if the birds were displaying any severe signs of heat stress, such as wing flapping, they were immediately removed from the box and basically released, allowed to cool and then released. So I used broken stick regressions in order to identify inflection points or a sudden change in each of these physiological traits, which indicates basically the onset of heat stress. And then I used linear mixed effect models to examine the effect of body mass and air temperature on each of these traits. So overall, we found that thick-billed murres displayed hyperthermia and increased their body temperature at about 33.7 degrees Celsius, which is low relative to heat-tolerant species, but similar to another cold-adapted bird we're studying, the snow bunting. We also found that air temperature was best predicted by body temperature. So above the inflection point, as you can see, there was a steep increase in body temperature in murres with air temperature. But overall, murres displayed a very low mean maximum body temperature relative to other birds. So they displayed a very limited ability to cool themselves, limited ability to increase their body temperature, which is an important adaptation for conserving water in birds. In terms of resting metabolic rates, we found that murres increased their resting metabolic rates at about 29.9 degrees Celsius, which again is low in comparison to more heat tolerant species, but similar to several cold adapted birds, including the snow bunting, but also several species of penguins. And we found that overall murres displayed a greater increase in resting metabolic rate over a narrower temperature range than more heat tolerant species. So they experienced a very high, greater increase in resting metabolic rates over a very narrow temperature range. So the greatest temperature murres could endure was about 39 degrees, whereas more heat tolerant species could endure, and particularly desert species, could endure up to 45 to 50, over 50 degrees Celsius. Now, in terms of the best model for resting metabolic rate, we found that body mass, air temperature, and their interaction best predicted resting metabolic rate, and larger murres experienced a higher rate of increase in resting metabolic rate than smaller birds. So in order to show the interaction, I divided the birds into two groups based on the median. And as you can see, there's a steeper slope in resting metabolic rate for larger birds or birds in the larger size class versus smaller murres. Now, in terms of evaporative water loss, we found that murres increased their evaporative water loss rates at about 21.2 degrees Celsius, which is very low, even relative to the snow bunting, so a cold adapted bird. But this temperature is ecologically relevant. It's similar to the temperatures in which murres experienced heat stress at Coates Island. And overall, murres displayed a very low magnitude of increase in evaporative water loss, which suggests limited capacity to cool themselves and dissipate heat. So they were very limited in their ability to basically lose water, evaporate water. In terms of the best model, we found that similar to resting metabolic rate, body mass, air temperature, and their interaction best predicted evaporative water loss. But this time, smaller murres experienced a higher rate of increase in evaporative water loss rates than larger birds. So again, birds were divided into two mass categories based on the median. And as you can see, there was a steeper slope, higher rate of increase in evaporative water loss in the smaller category versus the larger birds. Now, in terms of evaporative cooling efficiency, we found that murres displayed the lowest evaporative cooling efficiency ever reported in birds to our knowledge. So based on the values we could find in the literature, they had the lowest. And murres actually decreased their evaporative cooling efficiencies at 31 degrees Celsius. So 31 degrees Celsius is a low temperature, even relative to the snow bunting. But this pattern is extremely unusual. So this pattern has not been seen before. And so by the birds decreasing, so most birds actually will increase their evaporative cooling efficiencies at higher temperatures, but murres actually got worse. So based on this graph, they were actually producing more heat metabolically than they were dissipating. And finally, no murres were able to dissipate more heat than they produced metabolically. So if murres were efficient at dissipating heat, they would have values above 1, but the maximum value we found in murres was 0.33. So they are very, very poor at dissipating heat. So in terms of the best model for evaporative cooling efficiency, we found that body mass and air temperature best predicted evaporative cooling efficiency, and larger murres had poorer evaporative cooling efficiency. So as you can see, evaporative cooling efficiency declined with increasing body mass of murres. In addition, I also compared the maximum air temperature that the birds were able to endure relative to their body mass, and found that, again, maximum air temperature also declined with increasing body mass. So it seems that larger murres are more vulnerable to heat stress. So in summary, murres have limited heat tolerance. I can only say this in comparison to desert birds, because this is one of the first studies that has looked at heat tolerance in Arctic birds. Most studies have actually focused on cold tolerance. And over a narrow temperature range, murres displayed limited ability to increase their body temperatures, which is important for conserving water, a higher magnitude of increase in resting metabolic rates, so they're expending more energy, and limited capacity to increase their evaporative water loss in order to cool themselves. Resting metabolic rates increased at a faster rate in larger birds, whereas evaporative water loss increased more rapidly in smaller birds. And as a result of this, larger birds have lower evaporative cooling efficiencies and are more vulnerable to heat stress. So I would just like to, that's the end of my talk, but I would just like to, I would like to thank everyone at Arcus for inviting me. And I would also like to acknowledge the communities that I lived with in the Inuvialuit settlement region, the Rogers family for hosting me for many summers, as well as the many beluga monitors that I worked with and our research teams. I'd like to thank my supervisors for my PhD, Dr. Lisa Lazzetto and Jim Roth, as well as my co-authors and committee members, the hunters and trappers communities of Inuvik, Tuktoyaktuk, and Palatuk for their support, my supervisors for my postdoc, Dr. Kyle Elliott and Grant Kilcrest, as well as my collaborators and co-authors, the research team for Coates Island, as well as Middleton Island for their assistance, as well as the community of Coral Harbor for their support for our murre work. And finally, I would like to thank you, and I would be more than happy to answer any of your questions.
Thank you, Emily. That was a great presentation. Thank you very much. We have about 3 minutes before the top of the hour, so I think what I'll do is let people know that Dr. Choy has offered to stay for another 10 to 15 minutes at the top of the hour to answer questions. But for those of you who need to leave now, I want to share some information that today's webinar will be available within the next few days as an archive at www.arcusresearchseminarseries.archive. And you can also visit the Arcus homepage for more information about Arcus, our programs, and upcoming Arctic related events. And we are always looking for ways to improve our seminar series, as well as looking for suggestions for future seminar topics and speakers. Please take a minute or two if you're leaving now to complete our short survey. A link to that is available right now in the chat window, and it's on your screen, and will also be included in a follow-up email that you'll receive once this seminar has been posted to our archive. And with that, I'm going to go to the questions and answers. As a reminder, if you're joining by Zoom, please type your questions into the chat window. Several people have already done that, and I will read them aloud. If you're joining by phone, you will need to raise your hand by pressing *9 and unmute yourself by pressing *6. So we'll go to the first question that was asked, and this is following the presentation about belugas. This is from Heidi Zamzow. She asked, could you clarify whether belugas are harvested commercially or only for subsistence? And she follows that with a related question, do other nations such as Russia or Japan hunt belugas commercially?
Well, the Beaufort Sea beluga population is within the Inuvialuit region is harvested exclusively for subsistence, so by the communities.
And a follow-up question about other nations, do you know?
I know that they are also harvested on their migration route, but that's also from communities in Alaska. I'm trying to think of the group, but another Inuit group. So, but no, I don't believe that they're harvested by Japan or other nations. So it's exclusively for subsistence.
Thank you.
Inupia, thank you.
Thank you. And then Martin Pilot asks, do you see, this is a continue with the belugas, do you see any correlation with diving depth and sea ice cover and or temperature?
Well, I haven't, so I haven't been doing any work on actually the telemetry data, dive temperature, dive depth and temperature, but that is a good, I believe right now there is a doctoral student at Fisheries and Oceans who is doing a bit more work on, but actually has tagged beluga whales and might be doing some work on environmental factors and temperature and diving. But no, unfortunately, I wasn't able to actually test out some of the physiology work with actual dive telemetry. But that's a good question.
Thank you. And then Heidi Zamzow asks again, I realize this is somewhat peripheral to your research question, but have you considered the role of non-human predators in influencing beluga diving and foraging behavior?
Non-human predators, yes. So, I mean, I think that definitely in terms of predators such as, killer whales, I don't believe, like at least when I worked in the Inuvialuit region, there hadn't been any encounters with killer whales as of yet, but definitely, diving is a way to evade predators, as well as diving under sea ice. So that's one of the protections of the whales. So yes, I definitely think that there is an advantage in terms of, of predators such as killer whales and likely polar bears to evade them based on their diving ability. But I mean, this wasn't something that I personally studied, but I do think that there is definitely an advantage to dive ability with that.
Thank you. I think moving on to questions about murres, again, Heidi Zamzow asks, how did you control for the differential effects of stress on the birds induced by experimental conditions and not related to temperature, for example, confinement?
So we monitored the birds throughout the experiment. So we basically monitored their behaviors, and for the most part, I mean, they basically would spend the experimental run and basically sitting in the corner. But any data or based on behavioral observations, any indication of stress, we would basically removed from the experiment. So we only measured the most stable resting metabolic rates wherein the birds were calm. So that was one way we controlled. We also looked at, if the birds are stressed, you can definitely, you can see it in the behavior, but you can also see it in their resting metabolic rate signals. So they'll be all over the place, very, very high. So we only took the most stable values for our measurements. And it was only during the times based on our video surveillance and our observations of the birds. So we basically took notes throughout the experiment on their behaviors when they were calm. So that's how we control for it.
Thank you. George Devoki asks, do you feel a population level response of a seabird species is a better monitor of climate-driven ecosystem change than a physiological response? So population versus physiological response?
So that's a good question. So yes, we're hoping to look at more of a population level response in terms of heat stress. So one of the kind of papers I'm currently working on is to look at actually the impacts of environmental factors on, like we know that in years where the birds die due to heat stress, they also experienced greater loss of eggs, obviously, and they also were impacted by mosquitoes. So we're hoping to look at these results at a larger scale in terms of the impacts of warmer temperatures on egg loss as well as mortalities in birds. So no, I agree. It's we'll look at a larger population response to see if it's ecologically significant in terms of the impacts.
Thank you. We had a couple kudos from folks. The animal doctor wrote, very nice work, Emily. Looking forward to reading the paper on murres. And Brian Fadley wrote, thank you for the excellent presentation and your fascinating work, from a former Middleton Islander. And Steve McLean from NOVA asks, body conditions of belugas did not appear to be different in 2014 when Arctic cod consumption was low. Did you collect environmental data to investigate why diet changes in 2014? Why diet changed in 2014? And do you have thoughts about climate and Arctic cod availability to belugas and impacts on body conditions?
Yes, so no, in terms of in 2014, in terms of the condition in the sea and girth, it didn't appear to be significantly low. In terms of blubber depth, which was another indice I looked at, it was. Sorry, what was the second part of the question?
Did you collect environmental data to investigate why diet changed in 2014? And do you have thoughts about climate and Arctic cod availability to belugas and impacts on body conditions?
So I didn't personally collect environmental data. We did, because I partnered with the Beaufort Regional Environmental Assessment Program, they did do a survey of basically biomass of fish at trawling stations in 2014 versus previous years. So we did compare our results to the relative biomass of cod and other prey. So we do know that 2014 was the lowest. But in terms of other factors, no, I didn't specifically look at other factors. I mean, I know that the open water period within the Beaufort, at least, was the open water period. The belugas basically entered the estuary at an earlier time in 2012 versus 2014. So there was a bit more, sea ice extent was greater. But in terms of the second question of Arctic cod, we know that, based on other studies, that Arctic cod is quite sensitive to increases in temperature and water temperature. And in 2014, we're not sure why they were low in biomass. I mean, there's some speculation that they could have gone deeper and maybe they weren't detected. If the temperatures, the water temperatures increase, I mean, there is concern that the cod could move more north, and then and there could also be other subarctic species that the beluga whales are kind of exposed to. But yes, we think that the warming temperatures could have a detrimental effect on cod, definitely.
Thank you. And then another question that came in separately is, what is the range of temperatures that murres experience on Coates Island? And related, are those temperatures, do you see those temperatures increasing? Is there any data about increasing temperatures?
So, the temperatures at Coates Island, or the air temperatures, are much lower than my experimental temperatures. So again, the maximum is usually around 22, 23 degrees Celsius. But another factor that we're hoping to take into consideration in future studies is the fact that the birds they're black. So, if the murres are on a side of the cliff where there's a lot of sun, so Tony Gaston has actually done measurements of the backs of murres and has found that they could heat up to about 46 degrees Celsius. So we're hoping to look at the operative temperature as well as microclimates that could impact the temperatures that birds actually experience despite the lower temperature of Coates Island. But we know that yes, there has been increases. It is getting warmer in Coates Island. We know that from not only sea ice changes, but also increases in mosquitoes during warm weather years, but not to the point of, my experimental conditions. It hasn't reached 30 degrees yet.
Thank you. It looks to me like are there any further questions? I see no more questions in the chat. And seeing none, then I think we're ready to close the webinar. And so on behalf of ARCIS, I want to thank everyone that joined us today and for the good questions and discussions we had in chat. And I want to especially thank Dr. Emily Choy for your excellent presentation. This was really interesting. And then a little bit of information again, today's webinar will be archived and available within the next few days at arcis.org research seminar series/archive. You'll be able to visit the ARCIS homepage for more information about our programs and upcoming Arctic related events. We are always, again, please take time to respond to the survey, and that has been linked again in the chat. We're looking for ways to improve this seminar series, as well as looking for suggestions for future topics and speakers. Please take a minute or two, and we appreciate your input. The survey link will also be provided with a follow-up email to all attendees that you'll receive once everything has been archived. With that, again, I want to thank Dr. Emily Choy. This was a really good presentation, and we wish everybody a good day and an excellent weekend. And this concludes our webinar.
You're very welcome. Thanks for having me. It's fun.
Thank you.
Mhm.